Hey, hi, everyone. Um, quick reminder before we start in with the lecture. This is an article summary week. So hopefully all of you are well on your way to picking an article to summarize. And that's due tonight at midnight as usual. And so on Friday, when you log in, go ahead and log in to the appropriate Zoom meeting to be able to participate in the share day. Okay? All right. Let's talk about angiogenesis. So the angiogenesis, it's an interesting word, right? So it implies the creation of something. What is it creation of? It is a creation of blood vessels. So as we've been discussing the last couple lectures, the types of pressures that are faced or challenges that are faced by tumors that are beginning to grow and expand very rapidly, one of the things that we've mentioned is that as you get a tumor that's getting larger and larger and larger, there is a greater demand by those tumor cells to have access, right? Access to fundamental life-sustaining things like nutrients and oxygen. Okay. So angiogenesis is the discussion about how tissues can stimulate the growth of new blood vessels. Okay. We last time talked about drawing parallels between a tumor and a cancer, right? A cancerous tumor being kind of treated by the surrounding tissue as a wound that is never fully healing, that is constantly undergoing this healing process. Right? And we said a lot of that has to do with the leakiness of tumor capillaries. We'll take a closer look at that today. And also the trickery, right, of how these cancer cells, through random mutations, learn to upregulate certain types of signals that trick the surrounding stroma to do their bidding. Right? And we said as part of the wound healing response that's initiated, things like the growth and the encouragement of new blood vessels take place. So that sort of was that analogy of, I said, if you think of these cancer cells as becoming increasingly rebellious, right, and independent, just like a rebellious teenager that still needs to depend upon his or her parents for support, even though they're really beginning to disrupt sort of the normal household dynamics, right? And the parents are the sort of confused and manipulated stroma, right, that start actually doing things that might not be the most productive in terms of how to deal with this change in cell behavior. All right, so let's talk about blood vessels. Cells in your body need to be close to capillaries, right? Because the capillaries represent where things like oxygen are being transported throughout your tissues. And so as the oxygen arrives at different tissues, right, it has to be distributed in the form of capillary beds. Remember, capillaries are super thin vessels. Oftentimes, well, most of the time, the walls of capillaries are no more than just one cell thick. And it's important because with just one thin layer of cells, right, that's how you can get the diffusion of things in and out of the blood into the surrounding tissue. So that's important for gas exchange, nutrients, removal of waste products. One thing that you probably don't think about is how crazy extensive your capillaries are throughout your tissues. Oftentimes, like in anatomy class or even in immunology, when we're studying things like our circulatory system, we tend to draw it as a cartoon where you have like an artery that comes along. It's now divided into a smaller arteriole, and then eventually it has like this sort of, it turns into small capillaries on one side, and that's like red, right? And then it's kind of joined up with the other side of the capillary bed, which is blue, and then those little venules then go off to form larger and larger veins. Right? And that's how we usually picture it. But the truth is, it's not this neat, tidy little thing like this, right? The reality is it's running all throughout your tissues. The truth is that a cell in your body is typically no more than 0.2 millimeters or 200 nanometers away from the closest capillary. And that's because that distance of about 200 nanometers is about as far as things like gases can just diffuse through your tissue. If you look at these examples here, so here is a cross-section of normal liver tissue. See, these are hepatocytes. Everywhere that you see a red dot, right, these are red blood cells, everywhere you see a red dot, that happens to be a capillary that was cut in cross-section as part of this image. Right? If you zoom in here, you can see, so this capillary, oftentimes you think about it, right? The blood vessels in this tissue, they're not all running parallel, right? They're going in many different directions. So if you cut a cross-section, you're going to have some that are sort of like round, right? That's like a typical cross-section of, you think of it like a blood vessel, right? And you might see some red dots inside there, right? But others, if the blood vessel is at an angle and you cut it, you would see more of like a sort of funny oblong kind of shape with maybe multiple red blood cells in there, because those are the red blood cells that were flowing through it at the time, right? So that's what you see here. Right, so here you see maybe like 6 or so red blood vessels in this space, right? Whereas something like over here, right, this capillary was probably cut pretty cleanly in cross section. So you just see one red blood cell. But look how extensive the capillary network would be. Look at muscle. Not surprisingly, right, our muscle cells, especially our skeletal muscle cells, they need a lot of oxygen. Right? So not surprisingly, they're going to be really close to capillaries. So these brown, larger sort of oval things are the individual muscle cells that make up our muscle fibers cut in cross section. And look how in between them are the capillaries, right? So each one of these, so the red blood cell is sort of stained brown here instead, right? But look how extensive the capillaries are, right? Each one of these muscle cells has immediate access, not just to one capillary, but potentially 3 or 4 or more capillaries, right, to provide the ability for enough oxygen to diffuse into the cell, right, to power our skeletal muscles. So that's true pretty much throughout our body, anywhere you have living cells. Cancer cells also need oxygen. Right? I mean, just because a cancer cell has gotten all messed up with its genotype, it still has to use oxygen to do aerobic respiration in order to generate ATPs to power all of its various cell pathways, right? So that means cancer cells also need to be in close proximity to capillaries. These are a couple of pretty cool microscopy images here. So here's a melanoma tumor, right, that's been cross-sectioned. And what you see here is this is the capillary, okay? And notice how the cancer cells that are immediately around the capillary look like they're growing, all right? They have a pretty robust looking shape. But once you get out beyond 85 micrometers from here, right, after this dotted line, these cancer cells are dying, because they don't have oxygen. Right? Look over here. This is a prostate cancer cell in a rat. Again, the way this cross-section was taken, the capillary happened to be going through at an angle, so that's why you kind of see it as this section of the capillary. But same thing, right? The cancer cells that are immediately around the capillary, or this capillary down here, or this capillary up here, right? Those are the cancer cells that are rapidly dividing and growing. Once you go beyond a certain boundary limit, right, a certain distance, in this case 110 micrometers is what's being shown here, the cancer cells die, right, because they don't have access to the oxygen to keep them growing. And they become necrotic, right? So cells that just die because of lack of oxygen, they don't undergo apoptosis, right? They just die because they can't sustain their normal metabolic functions, so they become necrotic. Right? So you can imagine then that this creates an environment where there is selective pressure placed on these cancer cells to come up with mutations that will help them get around this stumbling block. Right? Some of those mutations might be mutations in enzymes that are part of anaerobic respiration, like glycolysis, because remember, all of these cells, all the cells in our body, when they're burning glucose to make ATP, they start off anaerobically by doing glycolysis, right? And you only get a couple of crummy ATPs out of it, but still you're making a couple of ATPs. So if you have random mutations in your cells that really, really ramp up glycolysis, right? Even though each glucose molecule can still only give you ATP, if you're really, really working on a lot of glucose molecules more than usual, or if you're really emphasizing the glycolysis of those glucose molecules to make it far more accelerated, right? You end up making more ATPs just from glycolysis alone. And those are what we learned about at the very beginning of the semester as examples of changes in the normal metabolism in cancer cells, such as the Warburg effects, right? Where certain cancer cells could then get away with not being close to capillaries because they have learned through random mutations that are selected for by these types of hypoxic selective pressures, right? You're selecting for those cancer cells that are stumbling upon these Warburg mutations. But the truth is that doesn't happen in all cancers, right? Most of the time, the cancer cells would just simply be a lot happier if they could get more blood flow coming into the tumor to provide more extensive capillary network to deliver oxygen. So this is an example of what happens in an extreme case when you have a large tumor mass that has become necrotic, right? So in this case, you can see, so this is an adenocarcinoma, and in the middle of the tumor, so I should say, right, this whole thing is actually a big tumor, and this is now just looking at different regions within the tumor. You can see this is the adenocarcinoma, the epithelial cells that have become cancerous, right? And they're still surviving because they're surrounded by the stroma where capillaries are flowing through here, right? But the cells that are in the middle of this tumor mass, they've become too far away. They've been cut off from the supply of oxygen, right, as the tumor is getting bigger and bigger. And as a result, they die off and become necrotic. That's why it's very often the case that during surgery, when surgeons remove large solid tumor masses, it's really disgusting, right? Because a large part of the mass might actually be dead necrotic tissue in the middle. Right? Okay. So when we talk about the vasculature or the formation of blood vessels throughout our body in order to support and sustain all the needs of our tissues and our cells, right? How much of that is pre-planned and laid out as sort of a part of human evolution? In other words, you know, our body plan, right? We all have a right arm, a left arm, we have two eyes, right? A nose, all this stuff, right? But what about the stuff inside of our body, right? We all have a pair of lungs, a heart, etc. But what about the actual structure of our circulatory network? I think you all know a large part of that is also predetermined by, you know, evolution and genetics, right? So if you were to just think about the main blood vessels that are coming from the heart, for example, the pulmonary vessels that go to the lungs and back, the aorta, right, and where the main blood vessels branch, you know, in our arms or in our waist, right? That is all pre-planned, right? That's why human anatomy books, you can actually learn the names of all these blood vessels, right? Because everybody's supposed to have that, right? You're supposed to have an aorta. You're supposed to have superior and inferior vena cava, right? It's kind of like if you look at a map, a road map. So here's a map, you know, of two big cities, and the highways are all indicated on the map, right? Because this is all part of the urban planning that was done there, right? And so therefore, this structure of these major thoroughfares, big roads, freeways, whatever, right? It makes sense to be able to have them on a map like this. Right? However, if you were to zoom in really close to a particular neighborhood, right? Yeah, you'd still see the roads are kind of laid out, but you'd also see there are oftentimes these funny little roads that maybe aren't on the map, right? Because those roads were sort of improvised, right? At some point in time, that little road was added on in sort of an ad hoc kind of manner, right? Ad hoc meaning just sort of spur of the moment as needed, right? Like shortcuts, alleyways, things like that. So here's Lombard Street in San Francisco, right? An example of, it's kind of like this really crazy sort of compromise, sort of improvised way of putting this this road connecting these two neighborhoods, right? So the point is that the growth of new capillaries is often done in a very ad hoc way, right? It's not something that came predetermined in terms of the actual layout of where these capillaries need to be, right? Your arrangement of capillaries throughout your tissues, is uniquely yours, right? So even though everybody would have an extensive network of capillaries running through your arm, right? That specific layout of capillaries, is uniquely yours, right? And it'd be slightly different in somebody else's body. Right? So how does a growing tumor improvise, right, a capillary supply? How does it encourage capillaries to grow towards it? One of the most important factors is this vascular endothelial growth factor, or VEGF. We said that VEGF is released as part of the wound healing process. Normally, when you get a cut, you're going to have damage to your capillaries, right? And those capillaries then need to be cleaned up, maybe removed, so that new capillaries can grow into the brand new tissue that's now filling in the wound, right? And as part of healing. So VEGF signals are a big part of that. So when VEGF signals are released, right, by cells that are sort of undergoing this kind of wound healing response, certain cells that are the endothelial precursor cells that are made in the bone marrow and are circulating through your blood already, right, when they get to certain parts of tissue where you have a lot of VEGF signaling happen, right, these EPCs, these endothelial precursor cells will stop there. They will differentiate and turn into new endothelial cells. So these endothelial cells, again, these are the cells that make up the walls of your capillary. Look at this image. This is kind of a cool image, very high resolution. This is a cross-section of a capillary, right? So the space inside over here is the lumen, right? And what this is showing you is how ridiculously thin the wall of your capillary is, right? It's one cell layer thick. In this particular image, we see two cells. Here's one cell with its nucleus, right? But if you follow the cytoplasm of that one cell, right, it's coming over here, and then here you can see that's the edge of that first cell. Right? And then over here is also the edge of that first cell. This other part, right, is another endothelial cell that is sort of cut off because, again, when you cross-section something, right, you don't end up with something that's like perfect, right? So in this case, you got in the cross-section a big part of this endothelial cell, right? But the other endothelial cell that might have been sort of deeper in this image here, all that you got of that one was just this part of it, right, that connects over here, right? So literally, the wall of your capillary is just one cell thick, right? And all the stuff in your blood is flowing through that, right? So when we discuss the formation of new capillaries, we should also mention that you'll need the growth of new lymphatic vessels as well. Those of you who took immunology, I think you have an understanding that the lymphatic system, as represented by these green vessels here, is essentially draped over our circulatory system, right? You can see that the green lymph vessels don't actually directly connect with our capillary bed. They're really just sort of intertwined with it because the purpose of the lymphatic system here is to drain away fluid, excess material, debris from cells, things like that, from this tissue site, right? Because remember, from the capillaries, you're getting all kinds of leakage of fluids, nutrients, things like that. The surrounding cells in the tissue, the sort of tan-colored cells here, right? They're producing waste products, things like that. And a lot of that fluid then needs to be drained from the site. Otherwise, you'd constantly have the buildup of fluid in your tissues, which is your edema, right? The swelling of tissues due to insufficient drainage into your lymphatic system. So part of the growth of a new capillary bed also involves the growth of these lymph vessels. So again, that's also stimulated by VEGF, and it's also stimulated by other factors released in the wound healing process, such as this basic fibroblast growth factor. As a result, you can imagine the layout of capillaries surrounding a tumor that has learned how to attract the growth of new capillaries, a tumor that has stumbled upon mutations that allow it to trigger angiogenesis, right? That network of new capillaries is not going to be very orderly, right? I mean, the cancer cells themselves are all messed up. The tumor is this big, nasty mess, right? The capillaries that end up growing into it, right, themselves are very messy. So here is an image of, I think this is a mouse brain, right, that has a brain tumor in it. And this is the area of the brain tumor. What's cool about this image is what they've done is I think they've injected some sort of fluorescing dye into the circulatory system. And the way that they image this is you just see where the dye goes. So in other words, you see the circulatory system light up, and the rest of the tissues surrounding it, right, you don't see. So very clearly, you can see these large blood vessels that flow into the brain. So these would all be sort of part of the normal genetic layout of how the mouse brain is designed. But as they branch smaller and smaller and smaller to form these little wispy capillaries, right, that becomes more sort of unique to this particular tissue sample here. Up in the upper left is where the tumor is, and you can really see, right? You don't see the tumor cells. Again, all you see is the circulatory system that's being highlighted here. And you can see how extensive and how messy and haphazard the capillary growth is, right? But at the same time, it's kind of scary because you realize because these capillary growths are so extensive going in and out of the tumor, they are supplying, right, the cancer cells the much needed oxygen that the cancer cells demand in order to keep growing and the tumor getting bigger. We mentioned that the capillaries that are running throughout the tumor themselves are very messed up and different in physiology than normal capillaries, right? Normal capillaries in red over here, so panels A, 2 panels here, these are normal capillaries, okay? The green staining represents the endothelial cells. So those are the cells that are sort of the wall of the capillary, right? I mean, they don't stain very evenly, but you get the impression that you can see the shape of the capillaries based upon where these endothelial cells are staining. The red cells are a different kind of cells. These are what we call pericyte cells, not parasite cells, right? Pericyte, peri meaning surrounding. These cells surround the endothelial cells and give them structural support. After all, if the wall of your capillary is only one cell layer thick, I mean, how strong is that, right? You can imagine the blood flowing through it if it's just one cell. So these pericytes form sort of an outer layer that gives stability and some rigorous to the structure. It's kind of like a garden hose. I remember, when we first moved into our house in Kailua, I said, okay, I got to get like a couple garden hoses, right? And I think I went to like Target or something like that. I don't know. And they had these cheap hoses that were like, 30-foot long hoses, and they were only like $7 or something like that, right? And I'm like, yeah, okay, sure. But these are pretty cheap hoses. They're just very flexible rubber hoses. And literally, like, the wall is just the rubber part, right? If you were to like bend the hose, you could see it's just the rubber part. And sure enough, after a few months, my hoses developed aneurysms, right? Where parts of the hose would just bulge because they couldn't handle the pressure of the water inside. So I ended up having to throw them out. So then I went to Hardware Hawaii and, ended up buying these like expensive, like $40 hoses, right? And those have not only the rubber, but around them, they have this kind of mesh, little wire mesh, right? That's kind of part of the wall of the hose, right? When you go home, you can look and see what kind of hose you have, right? But that wire mesh helps keep the hose from bulging, right? So that's like the pericytes, right? By the way, I think, Hardware Hawaii, I probably still paid way more than I needed to. If I had ordered the same hoses on Amazon, maybe they would have been $20. But anyhow, right? So that's what the parasites do. And you can see that here. They hold the capillary together. Let's look at, so those are normal capillaries. Let's look at a capillary that is growing through a tumor. Look at the parasites, right? They've all kind of fallen off, right? So they cannot hold on to and maintain the integrity of the capillary. So as a result, the capillary is much thicker. You can tell that just by looking at the green, right? It's more swollen, and not surprisingly, it's going to be more leaky, right? So you're going to have blood leaking out of these capillaries. And again, right, that's part of the wound healing response, because the surrounding tissue says, oh, we're bleeding, right? We better turn on the wound healing response and start growing some more blood vessels and things like that, right? So the ironic thing, of course, is that that represents the surrounding stroma then being tricked into helping the tumor, continue growing. All right. So here's some thought questions. What limits the angiogenic signals from normal cells? In other words, hopefully, yeah, you get it, right? If you get a cut, you need to trigger the wound healing response. There will be angiogenic signals, and you get the growth of new capillaries into that site as you heal. But what about normal parts of your body that aren't damaged, right? How come capillaries don't just keep growing? How do they know when to stop, right? So there are several inhibitory mechanisms that can limit vascularization. These are 3 examples that we'll look at for the rest of today's lecture. One is that the presence of oxygen, right, lots of oxygen, can actually be a way for your tissues to know that whatever capillaries that they have, that's enough, right? If your cells are getting enough oxygen, you don't need to grow more capillaries. We already looked at an example of this. Oxygen can help inhibit certain transcription factors like HIF from actually turning on VEGF signaling. All right, so we'll look at that on the next slide. There are other inhibitor molecules that are produced by your blood vessels, such as this TSP1 that we'll take a look at, that actually inhibit the blood vessels themselves from growing more. And we will discuss, what we talked about before, and that is the role of the extracellular matrix. The extracellular matrix that surrounds things like all of our cells, and in particular forms the basement membrane of epithelial cells, how that not only forms an anchor point for our cells to be positioned physically properly, but it also represents a reservoir that prevents certain types of mitogenic signals and other factors like VEGF from actually reaching cells. Okay, let's look at this oxygenation thing. So remember, we learned about VHL, von Hippel-Lindau, as a tumor suppressor gene way back when we were learning about tumor suppressor genes. And we said that VHL works by turning over or eliminating the levels of HIF transcription factors in your cells. So the HIF1 is a transcription factor that, if there's plenty of oxygen around, right, this enzyme, this proline hydroxylase, goes to work on the HIF1 and adds these hydroxyl groups to it. That makes it the right shape for this tumor suppressor gene, VHL, to bind to it. And the VHL proceeds to cause the HIF factor to be polyubiquitinate, right? And these polyubiquitin signals ends up shuffling the HIF1 to the proteasome to be degraded. Right? But in the absence of oxygen, hypoxic conditions, right, you're not going to have the HIF1 acted on by the hydroxylase. So as a result, HIF1 is allowed to go to work, and it binds to the regulatory sites of certain genes like the VEGF gene, right? So as a result, under low oxygen conditions, your cells produce the VEGF protein, right? And that VEGF protein is the signal that stimulates, one of many signals that stimulates angiogenesis. Let's look at this example here. So here we have a tumor region on the bottom and the surrounding sort of peritumor, right? So this will be like the surrounding stroma, for example. And what I want you to notice is the tumor in this case, the cells are very dense, right? Because they're out of control, right? They're proliferating out of control. They're dividing faster and faster in this limited space where it starts off. And as the tumor begins to grow, it pushes up against the surrounding tissues. You can almost sense just by looking at these striations or stripes here that the surrounding tissue is being compressed. What does that do? It ends up, right, collapsing the lymph vessels, sort of tearing them up, right? And it also tears up surrounding capillaries. Okay? So as a result, you're going to have, again, this is all part of the wound healing kind of response, right? Damage to surrounding tissues, but you're also, causing the blood to leak more, and you're interfering with the ability of these lymph vessels to properly function. So I think in this case, you end up getting a lot more drainage, rapid drainage of fluid from the site, because these lymph vessels are all tattered, right? So they're taking in much more of the fluid, right? And that can contribute to metastases if cancer cells end up in that fluid and get carried away by the lymph fluid here, right? So you end up with high hydrostatic pressure inside the tumor. In other words, the pressure of water inside the tumor is very high, right? And that means the water tends to want to flow out from the tumor as it's being squeezed. Right? So here's an interesting question. How might this impact the drug treatment of this tumor? Let's say the patient is given some sort of chemotherapy drug systemically in their body, so the drug flows through their circulatory system, and they're hoping that some of it will arrive at the tumor, right? But because of these high hydrostatic conditions of the tumor, where the water from the tumor is being squeezed out, this might actually create a situation where it discourages the soluble drugs that are sort of arriving at the tumor from actually getting into the tumor, right? So that's one consideration when you have a large solid tumor, that it might actually resist certain chemotherapeutic agents coming into it because of this environment of high hydrostatic pressure. Another example of inhibitory mechanisms are certain molecules that are released by things like the blood vessels themselves. So here's an example of a region of a blood vessel that has grown, right? So now we have this healthy formation of endothelial cells. They are one cell layer thick, sort of wrapping around, surrounded by those nice pericytes, right, in blue here, helping to keep it together so that as the blood flows through here, this thing doesn't bulge. Any one of these endothelial cells, you can think about it. It could either be an endothelial cell that's a mature endothelial cell that's already part of like the middle of the length of this blood vessel, in which case we can call it a mature endothelial cell. Or it might be an endothelial cell that's at the end of a capillary, right? That's growing forward, right? And as it's growing forward, these endothelial cells that are slowly forming at the very tip would be recently formed endothelial cells. So one way that our tissues limit the growth of these new capillaries is that the recently formed endothelial cells express FAS receptor on their surface. So FAS is the death ligand, right? So if FAS is stimulated, these cells undergo apoptosis. So the mature endothelial cells along the way, they are expressing TSP1. And TSP1 can be received by the newly formed endothelial cells as well as the mature endothelial cells, and it stimulates the cells to make the FAS ligand. Right? The mature endothelial cells, they start making the ligand, but they don't have the receptor for the ligand, so the cell keeps surviving, right? So the middle of your blood vessel doesn't just die off, right? Whereas the cells at the tip, if they're receiving the TSP1 and they make the FAS-L, they sort of signal themselves in this autocrine loop and stimulate apoptosis. So in other words, if there's no need for this blood vessel to continue growing, then the TSP signals, right, that are being released by the mature endothelial cells will limit the ends of the capillaries from growing further. The third example is the fact that you'll have this extracellular matrix surrounding the tumor, especially in the case of epithelial tumors, where you're going to have the basement membrane that the tumor is kind of pushing up against. And that basement membrane is going to have a lot of different growth factors and things trapped in it. So look at this image. This is one of my favorite images here, because this is a epithelial carcinoma, right, that's growing. So now, by now, the cancer cell, the tumor is still small, right? So this would not be detectable necessarily by X-ray, because it's maybe, what, a few thousand cells here in the middle. Right? But you can see these few thousand cells are beginning to really get crowded. You can barely make out the sort of light purple sort of line around it. That's the basement membrane, right? So these cells are really shoving up against the basement membrane, stretching the basement membrane out. Right? What is this brown thing? This brown thing, right, is a capillary, a nearby capillary that was kind of just flowing by over here that has now been tricked to grow towards the developing tumor, because these tumor cells have learned, right, through random happenstance, randomly stumbling upon a combination of mutations that are allowing for things like VEGF signals to be produced, right? So the VEGF signals kind of float out from these cells in a cloud, and when they reached this capillary over here, they stimulated then the growth of the capillary in this direction. And now look, the capillary is now growing all around the tumor, right? So what will happen now is, yeah, you better believe it. Now that this little tumor has a pretty extensive capillary supply forming around it, suddenly the growth and proliferation rate of these tumor cells is going to start ramping up, right? But importantly, what? They're still contained by the basement membrane. So the next critical thing that we're going to talk about is what happens once that basement membrane is disrupted. Right? Oh boy, you can already see where this is going, right? So tumors must come up with, through random mutations, the right combination of mutations to suddenly make what we call this angiogenic switch, right? The size of the tumor is beginning to be limited, such that the cells in the middle are beginning to stress out because they're too far away from a blood supply. So now they're learning how to get a blood supply to come to it, right? That's part of this angiogenic switch. And once that angiogenic switch happens, and all the tumor cells are happy getting plenty of oxygen, and they really start expanding, now the integrity of the basement membrane that's holding them together is now at risk, right? So the important thing to consider is that limiting vascularization through those different mechanisms that we talked about, those three examples today, is yet another way that we have evolved to put the brakes on tumor formation, right? So not only inside of the cells do we have tumor suppressor genes, but on the outside, we have these different ways of limiting the growth of things like blood vessels to discourage tumor lesions from really kind of growing too quickly. Right? So tumors must break through the basement membrane in order to really start manipulating the stroma even further, to really start encouraging a lot more capillary growth. So here's an example. If you look up here, here's normal skin, right? So here's the layer of our dermal cells, our keratinocytes, et cetera. And then beneath the dotted line, you can think of as kind of this basement membrane. Right beneath them is the connective tissue, the stromal tissue. So stained in red is where you have the capillaries that happen to be running through this image, right? Most of them are in cross-section. During hyperplasia, so now the skin is becoming hyperplastic, you still have the basement membrane holding up. But what is happening is some of these hyperplastic cells are already learning how to encourage more blood supply, right? So it looks like, it's not a dramatic change from normal skin, but you begin to get the sense that there's increased capillary growth here, increased vasculature. Eventually, when you get to the point where you're really talking about dysplastic growth, right? Now those tumor cells are really losing their shape, right? You can really see they've now learned how to really fool the surrounding tissue, right, to supply them more and more capillaries. And now the basement membrane itself is threatened, right? The integrity of that barrier that's keeping these tumor cells from spreading. Right. So we mentioned how certain immune cells that are part of the stroma also end up being recruited or fooled into helping out with the tumor growth. So in this cartoon here, if we look at when the tumor is still in situ, right? So meaning these are little pockets of tumor cells that are growing here, but they're still kind of contained within their basement membranes, right? So this is a hyperplastic islet that's happening in something like maybe a pancreas, for example. Right? Certain steps that might happen is there might be certain cytokine messages that are released by these cells that begin to fool the stroma around it, and in particular things like mast cells or the immune cells that are first formed in our bone marrow and then go to work in our tissues like mast cells and macrophages, right? They might come up to this tumor and believe that it's a wound, and so they may secrete things like those different metalloproteinases, matrix metalloproteinases like MMP9. So what happens is you have things like this is a molecule of VEGF, the gray thing, right? So this is a molecule of VEGF, it's a protein, but it might have been sequestered or held by all the fibrous proteins that are in the extracellular matrix, right? And as a result, this protein never made it to the surface of the tumor cells. It's just been held by the basement membrane. But the matrix metalloproteinases that are being secreted by these immune cells, again, because these immune cells think they're doing their normal job. Their normal job is we have a wound here. We need to kind of clean up the mess here. So one of the things we're going to do is digest away the extracellular matrix so that we can clean things up to allow brand new cells to grow. But in digesting away the extracellular matrix or the basement membrane, they release all those types of mitogenic factors and angiogenic factors that were held in there. So as a result, right, you then suddenly get increased growth here. So this islet now has much more capillaries supplying it, right? These red things were meant to be the capillaries that were flowing through here, right? So that can happen through this more gradual process of the wound healing, right? Or it could happen if it stumbles upon the angiogenic switch very quickly, right? For example, upregulation of VEGF. And if you suddenly have a ton of VEGF just being made by these cells themselves, right, that can also do the angiogenic switch. And that will then encourage the cells to now break free of the basement membrane and spread. So this is the last slide today. So I mentioned that as the tumor is pushing up against something like its basement membrane, right, and it's beginning to struggle because it's getting kind of big, so the cells in the middle of it are beginning to get stressed out because they're not getting enough oxygen. So maybe, right, it stumbles upon certain mutations that gets more capillaries surrounding it, right? But still, right, the ability of VEGF to even float out from the tumor and get into the surrounding tissue is limited by an intact basement membrane. Right? We talked about the basement membrane as being something that's preventing mitogenic signals from arriving at the epithelial tumor cell, but likewise, right, the basement membrane works two ways. It'll also limit the ability of things like VEGF to be able to get out of the tumor into the surrounding tissue. Right? So up until this point, if you have an intact basement membrane, right, the tumor is still struggling to make this angiogenic switch happen. But once that basement membrane breaks down, right, and all those things like mitogenic factors can now arrive and reach the tumor cells, stimulating them to grow even faster, and all the VEGF that those tumor cells were releasing can now make it into the surrounding tumor. You can imagine angiogenesis will happen much more rapidly as well. We can see that in these nice photos here. On the left is prostate cancer. So this is when it is still prostatic intraepithelial neoplasia, right, or PIN, right? So this is when the prostate cancer is still in situ. So in situ means it's still in the place where it started from. You can think about it as at the site where the tumor first formed, right? And you can notice, what do you see? You see that in this particular cross-section, you have a lot of necrosis already happening. So these cells in the middle were really stressed out and died, right? But you have a lot of cells on the outside here that are still rapidly dividing, and they're still contained by the basement membrane. That is why the capillaries that have been sort of encouraged to grow towards them, right? They're sort of still stuck on the outside of the tumor because they can't break in because of the basement membrane. But once you lose the basement membrane and the basement membrane is digested away, then look, right? Then it just becomes all crazy, right? Because now you have the capillaries can grow right up against the tumor cells. The tumor cells have access to all these mitogenic factors, right? Things are just going nuts. Right? And at this point, it is no longer in situ. Now it is invasive. Right? Once the carcinoma has broken out of the basement membrane and starts infiltrating the surrounding stroma, it is now full-blown cancer, right? On the bottom, same thing, right? Here's breast cancer in situ, right? Where you can tell the middle part, you have some necrotic parts of the tumor because these were too far away from the blood supply. These darker pink ones are the rapidly dividing cancer cells, and these dark red spots are the capillaries, right? So it's amazing, right? Look at it. You can really tell they've managed to start getting capillaries to grow towards them, but they are still separated from the outside by the basement membrane. So the capillaries can't get in and the tumor cells can't get out, right? But once you lose that basement membrane, right, it just becomes a big mess, right? And needless to say, once you've made that transition in the tumor from in situ to invasive, then the prognosis for the disease is dramatically going downhill for the patient. Right? All right, so let's go ahead and stop there today.